The first step of turning a gene into a protein is called translation. There is a region of the DNA called the data box. Here, a transcription factor binds to increase the DNA affinity for RNA polymerase II. Once bound, the RNA polymerase unzips the double helix. When it reaches the start point on the DNA, it begins transcribing the template strand. RNA polymerase adds nucleotides to the 3' end of the growing RNA molecule as it continues along the double helix. The sequence terminates shortly after RNA polymerase II transcribes a polyadenylation signal. Before the pre-mRNA can be exported from the nucleus to the cytoplasm, it must undergo a series of modifications called RNA processing. This processing occurs in the nucleus. There are segments on the pre-mRNA called introns and exons. Introns are non-coding segments of nucleic acid, and exons are the region that are eventually expressed. Before introns and exons are dealt with, the 5' end of the mRNA is given a 5' cap, a modified form of a guanine nucleotide. After, the 3' end receives a poly A tail, a strand of 50 to 250 adenine nucleotides. Once these modifications have been added, introns and exons are ready to be spliced. Pre-mRNA is spliced by a spliceosome, which is composed of small nuclear ribonucleoproteins, SNRNPs, bound to other proteins. These spliceosomes interact with certain sites along an intron, releasing the intron and joining together the exons. Once the RNA splicing process is complete, the new modified mRNA is ready to be exported out of the nucleus. The mRNA leaves through an opening in the nuclear envelope called the nuclear pore. Although the 5' untranslated region and 3' untranslated region are not translated, they are not spliced out of the mRNA because they serve other functions such as ribosome binding. Translation has 3 steps: initiation, elongation, and termination. During the first step, initiation, the small ribosomal subunit attaches to the messenger RNA. The transfer RNA has an amino acid binded to its top and has a nucleotide triplet called an anticodon, which is what allows the transfer RNA to bind to the messenger RNA's nucleotide triplet, referred to as a codon. Once the tRNA binds to the mRNA, the large ribosomal subunit comes and binds to the messenger RNA. The union of mRNA, the start codon, methionine, which sits in the P site, and the large and small subunits of the ribosome completes the translation initiation complex. In step 2 of translation, elongation, the ribosome moves down the mRNA like ticker tape in the 5' to 3' direction. In this stage, amino acids are added one by one to the previous amino acid at the carboxyl end by peptide bonds. tRNA enters the ribosome through the A site and leaves through the E site. This occurs until the polypeptide is completed. Termination, the final stage of translation, occurs when a stop codon reaches the A site. A release factor binds to the stop codon, releasing a water molecule that hydrolyzes the final polypeptide tRNA bond. The resulting polypeptide sequence is methionine, lysine, phenylalanine, glycine, with methionine at the amino end, also known as the N-terminus, and glycine at the carboxyl end, also called the C-terminus.